Sándor, Pongor.
Okay.
I had a class with him.
Great environment to be in Hungary and Budapest. I have never been to Budapest. Okay, so today we continue the theme of 1D predictions, this particular case of helical membrane predictions. We are going to have next Thursday a talk from Thomas Hopf about contacts, and we have an interjection from Professor Bromberg from Rost University that will be next Tuesday. Begin with membrane again. So membrane, in some sense, you have to create a wall between self and the environment. What do you put in there, in the wall?
Transportation mechanisms, tubes.
That's an interesting idea. This is certainly when you build a house, right? You have all these tubes going through the wall. So you put doors.
Receptors.
Receptors.
Signaling in output.
So if we, in some sense, my image is always you have a town, a medieval town or something like that, and build a wall around it. So we have the receptor idea, we have the idea of doors, we have the idea of tubes. Could you find analogy to these ideas in a cell? So this, I believe, cell wall is the wall around the medieval town. That's trivial. What would be the receptor? What would be the door? Any word for that? In a cell?
I think I don't get a question, right?
So in the medieval town, you have a wall around it to protect the inside and keep the outside in, and then you have a cell. In the cell, you have a cell wall too. In the cell wall, then the image of the house or town got to the words of door or gates. The image of the house or town got to tubes. And the image of a cell got to a receptor. But I'm trying to sort of find the corresponding points in these two images. So what would be the receptors in the town? And what would be the gates and the tubes in the cell?
Are we talking about the receptors on the outside wall of the town or in the town?
Which one did you talk about?
Inside the town, also every house has like its own wall because like it's compartmented inside.
So let's look at the. Is that true for everything?
No,
every cell?
Some
every.
houses.
Let's look at a bacterial cell.
Right.
Do they have inside houses in that image?
Mitochondria? It's like.
Mitochondria is not in a bacterium. Mitochondria is or was a bacterium, but bacteria, no. Well, now we are talking biology. Don't push me with is there any exception? Yes, sure there is. But typically, the idea of a bacterium is that you have a wall and something inside and something outside. As simple as that. And you have no structure inside in principle. Yes, you have some structure, but you don't have a nucleus. You don't have compartmentalized membranes inside. You don't, in that image of a house, you may argue in a eukaryote you have mitochondria, there could be a house in a town. That's a very, very good image. And the mitochondrium has also a wall around it, so the image is completely right. But for the bacterium, that is not the case. But again, my question is, your image of a receptor at the, let's talk about a bacterium at the outside membrane. What would that be in the image of a town?
Could be in the metaphoric way. Metaphoric way.
Yeah.
Could be a camera. Could be a.
Oh, that's a good one.
Could be a button on the wall.
Yeah, that's true.
Could be like whatever.
That's a very, very good point. There is, however, one important thing here. Most of the time, signaling is transmission of material. So your image of the camera, well, typically you have the camera to have somebody let somebody else in, true. But the camera itself is only transmitting the image, not the material in some sense.
The camera opens the door. The button opens the door. So you press a button and then the button checks
Great.
the fingerprint and then opens the door if you're on the right.
100% with you. That's a great. That's a great image, and that exactly is what a receptor could do. Yes. Now the receptor would also do sort of the. Well, my point is the receptor is in some sense also the gate, right? Because the receptor is in fact sensing it as the camera, plus is the entire mechanism. In your image is the entire opening mechanism, in camera included. That's a great image. I never thought about that. I like it. So but the point here is the gating mechanism in some sense are the receptors. Back to the tube. So the doors are the receptors, and that is in fact, so the two of you talked about the same thing essentially. What about the tubes? So in houses we have them to get some stuff out and some stuff in. Water and other things, electricity. What is that in a cell? Do we have exchange? Is there material that comes in and out? Do you know any creature is the wrong word, anything that certainly consists of DNA that does not have a constant exchange? What I mean is a virus. A virus essentially has a shell around it, or whatever you call it, that has to be cracked open essentially once to inject that material, the genomic material, into a cell, into a host that will then reproduce that virus. But it does not have any constant flow. And that is in principle one of the big things that distinguishes living creatures, cells, from everything else. We have a constant exchange. Back to the question. Pipes that go in and out. What would they be in a cell? Are there pipes? Just what are the pipes? So there are. The image of a pipe is not misleading, but you would typically call them something else. Microtubules. Oh, that's a great idea. The microtubules are a little bit more. My analogy for those would be more train tracks. Because you really, on the microtubules, you imagine like a cell completely full of spider webbing tracks. And material is transmitted or moved around in a cell on these microtubules. And that's why I call them train tracks. But they're active train tracks. They're train tracks in the sense that they also have the transport mechanism to advance the train. So the train track is the image, the train moves, and the track is just there. Microtubules are in fact active. The actin is moving things ahead, myosin. So that is not the tube outside typically.
Well, they are like classic beta barrel tubes, and
yes, porins.
So they are like
exactly.
Carrier proteins, like changing things. Yes. So there are many ways of.
Essentially, we have proteins that are at the interface, and that, in fact, we have tubes that are more or less active. We will talk about that on Thursday. The porin type of non-active transport, essentially holes, essentially pipes, and more active regulation. Now, my question to you again is. What can you, if you had to invent a cell, what could you put around it? So you need some material that allows you to have exchange, that allows you to have an inside and an outside. What type of material would you put in there? So you all know what the material is that life chose, right? What is this membrane made of? Lipids. It's an oiled phosphates, but essentially the backbone in some sense is really the point, the biophysical point essentially is a layer of oily substance, a lipid. And why is that so important? Why do you put a lipid? Because inside essentially you have water, outside you have water. You have a drop of oil swim on water, and you drop oil on water and you immediately see those two don't mix. And that don't mix translates to you can keep inside what is inside. right? And at the same time, you have something that is absolutely mobile, but it's not like concrete. It's not like, and that is what the cell achieves. Now, you could have achieved that with proteins. We got to the point that the tubes, the gates, all of these are proteins. You could imagine that you built the entire wall by proteins. Why do you believe that has not been done? Or can you think about a downside?
I think it's the most practical way, like the most easiest way. Like they are so complex.
Yes. Well, how would you define easy? Typically, it's a dollar thing in some sense, right? It's cheaper to get lipids around there is relatively cheap in terms of energy production. If you would have to build the whole thing by proteins that have the right structure, that's a lot of organic material that you have to first assemble, produce. Immense amount of energy. I assume that is the reason why this is not the case. You could produce something that, in fact, could possibly do the function of a membrane even better than a lipid if you produce the whole thing by proteins. Maybe there is a life form that does it, and then viruses sometimes have shields like that. But the cheapest way is the lipid. 2 movies about that. That brings me to the point where I forgot to plug in. But you guys are, no, we're not going to do movies. You're too far ahead. So again, the point about a membrane is in a normal protein, you have inside an oily environment in some sense, outside in a water environment, so hydrophobic inside, hydrophilic, hydrowaterphilic friend, phobic enemy. Inside you want to be you are enemy of water. Outside you are friend of water because it's dissolved. The lipid bilayer creates an environment, by the way, about 3 nanometers thick. Creates an environment with the 2 lipid groups pointing toward each other that is completely hydrophobic. And that leads to an inversion of the typical. Now instead of having around you solvent that is where you want hydrophilicity, you now have an environment where you want hydrophobicity. So essentially the protein sitting in the membrane looks like a protein inside, or the part of the protein that usually it sits inside of a protein. Yes. And there are 2 classes of transmembrane proteins. One go through the membrane with alpha helices, shown here. On Thursday I will talk about the ones that cross the membrane. You alluded to them, porins, that go through the membrane with beta strands. You do sort of see in these helices, in these rods, and those are different proteins, you see an assembly of proteins, of helices, where you sort of, in this case, see that it's like a mushroom up here and squeezed. The squeezed part is the membrane. Here too. Here the membrane and the outside, or the outside of the membrane. A couple of other examples. Again, all of these. Here you do have a channel. That's in fact a mechanical sensor, where you sort of see the hole here through which you can transport material. Again, some of these membrane helices may be anchored, like this blue one here, meaning they don't go through the membrane, they just sit there and in some sense hold onto the membrane. And in this particular case you see one that is holding and going through. In some cases you have membrane helices that, when you look closer here at the red one, part of this red one here goes through the membrane as a membrane helix. Part without much of a kink here continues in the globular region. The same is true up here. So some of these membrane helices sort of have ends to them. In this particular case, there's a part that is anchored and a part that is anchored in some other globular part of that protein. Mostly, however, what you see here again, you see sort of things that go in and out. You see extreme kinks here, but mostly what you see is helices that in many ways are orthogonal, largely orthogonal to the membrane. So if the lipid bilayer runs here, then most of these somehow, now I picked an example here where that is not the case, but most of these are pretty close to orthogonal through. When we do comparative modeling, I showed you on the Swisspot view, it looks like this. On the genome view, our ability to do comparative modeling looks a little bit like that. But over time, our ability to do comparative modeling increases here simply because the database grows. For membrane proteins, this is what you can do comparative modeling with. And over time, you see there's not much of an improvement. Why is that? Why is comparative modeling so unsuccessful for membrane proteins?
Because we can't get the exact structure from experiments.
The problem is, exactly, you're right. So the problem is comparative modeling is about mapping to known structures. We don't have many known structures for membrane proteins. In fact, overall, this is done a few years ago. Today we have about 100,000 structures in the PDB, and it still is true that fewer than 2% of all the known structures are membrane proteins, at least the unique one. And back to your country here, Shoshana. But over 20% of all the proteins, and I'll get back to that number, are in fact have helical membrane, have membrane helices. Very few of the known structures, many of the proteins we have. Now, essentially what we want to have, at least, so we want to have 3D structures, but that is a complication now. But at least what we want to have is we want to know where the helix sits. We want to know how many helices we have. For membrane proteins, the number of helices is very important for the function. Different types of proteins are often recognized by the different types of helices, number of helices. For globular proteins, there is no such thing. You can build up seven regular helices, globin, you add a helix, you do something else, you can actually have globins that have not 7 helices. This I shouldn't have on YouTube. I'm not entirely sure it's true. But almost every, even for a tin barrel, there are examples where we don't have the regular tin barrel. So the number is clearly not as revealing as for membrane proteins. For membrane proteins, we have 70 M proteins, the GPCRs, G-coupled receptors. It's a class of proteins that is of which you find a lot in mammals. Hundreds of those in human, just for smell alone, and that is a particular class you don't find in most bacteria. Bacteria have transporters that have 6 or 12 membrane helices, which you don't find in eukaryotes. So you have really different universes. They solve the problem of importing, exporting stuff in very different ways, and that you can see on the number of just seeing the helices and the orientation. So where the protein begins, where it ends, that's another aspect of it. Another reason why we, in fact, would very much like to have more knowledge about structures is when we look at drug targets. 57% of all the drug targets on the market are membrane proteins. The others, some of these others are also membrane proteins, but internal membrane. This is external membrane, but some of these in fact are also membrane proteins. So we have a high incentive to get structures for membrane proteins, and we have very little structure, and that pushes, for instance, about almost 40% are just targeting one particular class, GPCRs, I talked about them, 7 transmembrane helices shown here. So we want to get there's another one of channels and pumps. How can we predict membrane helices? So the cheapest way in which we can do that is we simply take what we already have. So we take a method that predicts helices. A secondary structure prediction method, we talked about it, right? Would that work? As a helix. Helix is a helix. I show an observed transmembrane helix, so that's the sequence, and that's the prediction. Utterly wrong. Well, not utterly wrong. There's some residues that are right. But you know, this is as bad as it gets in terms of making prediction mistakes. Why is it so wrong? Because the environment is different. The environment in which you form a membrane helix. So if you did not have lipid, maybe this would form a beta strand. And if you had a coupled strand to back it up. Yeah?
What if I say search for helix with hydrophobic attributes?
Yeah. So you mean in globular proteins, or?
Sorry?
You mean in globular proteins? So if you could.
No, in membrane proteins that are, say, find helices that have the attribute of being inside them.
But in this particular case, this would not be predicted as a helix, so your algorithm would fail right there, right? Because you say it should be helix and should be hydrophobic. And here you would not have the prediction of helix. So your algorithm would never do the second step, at least the way you formulated it. You could also turn that around and say, look for hydrophobicity first and then forget about membrane helices. Actually forget about any helices and just look at hydrophobicity, but then you completely turn around your idea of an algorithm. Yeah, that's the point, that he would fail here, right? Oh, you mean both? You mean either or and, a logical?
Well, I would say and.
Yeah, that's the point, he would fail, right? Because you already have the E. Well, and means both. And this is predicted to be
right,
a beta.
But can't I not do this one, but...
A new method.
Yeah, new method, right, yeah.
Good, we get into that,
Fine.
exactly. And so again, that's the reason, and you're totally right. So we should begin with looking for hydrophobicity. How would I look for hydrophobicity? Well, one way, so this is David Eisenberg here, who brought us 31 years ago, one of the scales for hydrophobicity, where you see for every single residue the degree to which it's hydrophobic, which they did as a mixture of an experiment and computational method. By the way, David is one of the pioneers, early experimentalists who really pushed computational biology in many ways. Isoleucine here, phenylalanine, valine are highest on the scale, and arginine, so the positively charged residues, negatively charged residues, on the lowest scale, on the low part of the scale. Very hydrophobic, very hydrophilic. Now, if I showed you, this is the scale from David Eisenberg. If I showed you another scale, would that be identical? So let's get back to what I glossed over. Those side chains often are considered hydrophobic by some. I mean, here I have a scalar. Here I just say, well, somewhere I make a cutoff here, and in that cutoff, everything else is hydrophobic above, and that's what I colored green here. In fact, exactly what I just showed. Yes, exactly here. But if I showed you the next hydrophobicity scale, would it be the same essentially? Let's not worry about the numbers, the very numbers.
How did he do the index using only chemistry?
Well, it was a mixture, so between experiment and computation.
Experiment of what?
Essentially, what you try to do is take the side chain out of solvent, the energy. Try to measure the energy of that.
Sorry, what was the question?
Hydrophobicity. Your idea was to use hydrophobicity. And here I have a hydrophobicity scale. But now, since I have ample time in my lecture, I'm idling and I'm coming up with a question that is not really helping us to predict our membrane proteins, but it's slightly misleading, namely the question, I have one, and I should just declare victory and move on. Or I could ask, is there another one? And if I found another one, my question to you would be, I'm going to present you another one in a minute, or less than a minute. What do you expect? Will it look the same?
I would say no, but I don't know why.
You have learned on a social scale. You have learned too much already from observing me. Well, the semester is over soon. You can get a new teacher. So here we have the Eisenberg scale in green, and this is an information content. Negative is hydrophilic, positive is hydrophobic. Kite Doolittle in blue, and another. All of these are essentially experimental scales with slightly different ways to do the experiment. Now, there are two ways of looking at that. There's a high agreement, essentially between them, or you could say they all differ. Let's just see what happens if I look at more. A few more numbers. Now we get into numbers that are slightly more theoretical or slightly different. Japanese computational biologist, by the way, is measuring how much of your environment is surrounded by other side chains, or could be occupied by water. In that sense, it measures a similar feature. Gunnar von Heine, we'll get back to Gunnar in a minute. That is something that is partially done. At least the one that I show here is partially done, more done by computational concerns, really looking at membrane helices. But you begin to see that there's a variety. Now, if I clustered, and this is not I-96, done by a Japanese group, Kaneisha, to me, and Kaneisha, you see that they do cluster by scales that measure hydrophobicity. Some of these scales measure very, very different aspects of protein structure, such as propensity forming turns and so forth and so on. The point is that in the scale of doing something very different, beta strand propensity, turn propensity, this cluster here is fairly large. All of these are declared hydrophobicity indices that have been measured differently, have been computationally done differently. This is already 20 years ago. So there's a wide variety. And you want to put that in, the idea that you have is exactly the idea that is right, but it's actually not that trivial. Because there's a variety of hydrophobicity. Which one to use is in itself an issue. But let's go back to this issue and take, in this particular case, I cannot even remember. I assume I used the one from David Eisenberg. And I introduced an ad hoc threshold for calling a residue hydrophobic or not. And for this particular case here, you see that the H, a simple threshold in H, would predict very, very closely a membrane segment. So where you see the red H here, that essentially overlaps very well with the hydrophobic region. So the first part, or one part of your algorithm, seems to, in this particular case, already get you there almost. And that, in some sense, is the story or the way of doing it that has been pushed by Gunnar von Heine, shown here. One of the shapers and shakers in the field. H-index over 75, 7 papers cited over 1,000 times, 63 over 100 times, is in Stockholm University. And here he is shown as the chair of the Chemistry Nobel Prize Committee, announcing the 2008 Nobel Prize. So that's another one of those moments when you realize somebody has made an impact, and he's actually back into the committee as of last year. So here the idea for membrane prediction, and those are the early papers, essentially this is the idea. To use hydrophobicity in a clever way to predict membrane helices. At that time, you may remember I showed you Chow-Fassmann, I showed you methods that predict secondary structure. I showed you the Gore methods, the first series of the Gore methods in the beginning of the 80s. They predict secondary structure, and Gunnar goes and focuses on membrane proteins and really looks more at hydrophobicity than at helices. That's how the story evolved. Then he plots some hydrophobicity index here, and that in particular is his own. And now to assign membrane helices, you begin to simply put a threshold. Now in this threshold, everything above the threshold is a membrane helix. 1, 2, 3, 4, 5, 6, 7, 8, 9. Is that right? Well, the point is that you need to go through roughly 3 nanometers. 2.5 to 3.5 nanometers, the thickness of a lipid bilayer. You have to cross that. And you cannot cross that with a helix that has 5 residues. is just not able to span as a helix. You could pull it apart, right, like a spring. But as long as you maintain a helix with hydrogen bonds, you need more than 3 or 5 residues. In fact, what we know is that typically you need something in the ballpark of, if you go orthogonal through the membrane, in the ballpark of 17 residues, and that leads to a situation where some of those here you simply will remove because they're too short. They cannot cross the membrane. And again, I should have shown that picture. That's the thickness of a membrane. So in this particular case, you may end up with just 2 membrane helices predicted. Now here comes the next argument of Gunnar. He says, okay, these are clearly 2 membrane helices, but it's also true that there are areas here, regions here, that are very high in terms of hydrophobicity, and they are extended. So clearly there are examples here where you have a peak. So that most likely has nothing to do with the membrane. But there are areas that may not really make it to 17. And already visually you're guided into the next idea. The next idea is to have a second threshold. To argue you have to have a certain minimum in order to ever sort of insert part of yourself as a membrane helix. But in really to span, you can maybe at the ends or in the middle, you can afford to have a little bit less hydrophobicity. You can have flanking regions that are a little bit less hydrophobic as long as the bulk of the membrane helix is. So as long as the second threshold tethers out, and that still sums up to 17 or something like that in this particular case, we would, in my example here, we would accept 5 membrane helices. So the first 2 come in through the first threshold, and the other 2, because by extending them, so there's part of that that falls under the first threshold, so they have sort of the trigger mechanism of insertion. And then there's an extension, as long as you don't fall below some threshold, when really the signal gets too low. As long as that is the case, you in fact accept that as a membrane helix. So these 2 are the first, the other 3 are the second. There is some more clever idea of how to sum in fact over the hydrophobicity that he introduced. I gloss over that and I bring a new aspect. So far, what that method would give you is to tell you essentially that is a membrane helix, that is a membrane helix, that is a membrane helix. I said the number of helices is important for the function. I also said that the orientation is important. So far what I told you will not give you an orientation, right? How could you get the orientation? So with orientation in the membrane field, that is typically misleadingly referred to as topology, is simply where is your N-terminus? Is your first residue in the protein starting in the outside of the cell or on the inside? Okay? And how could you predict that? So I gave you, so far the method gives you the number, what Gunnar showed you so far gives you the number, the membrane helix. How could you look at the other parts?
Probably the starting positions have different attributes depending on if they're in or outside. Maybe like different propensity of different amino acids.
What could it be? And again, this is nothing that anybody of you should know I'm not expecting that anybody knows the answer. But let me ask the question slightly differently. Do you believe that there is a different biophysical environment in the inside and the outside of a cell? Yes, we have slightly different pH values. In neurons, there's a gradient of charges, which leads to the firing of neurons, which leads to neurosignals, which leads to reaction and all kinds of other things. So yes, there clearly is a gradient. And now the only question remains, what kind of residue feature? And Gunnar showed in a very early paper in '86 what he coined the positive inside rule, the Gunnar von Heine rule, which is simply you sum over the, let's call them loops here. They're not really loops, but so the non-membrane part close to the membrane helix is here on this side and on this side. Blue, periplasmic, outside, cytosolic, inside. And all he shows in this one here is the positively charged residues, K and R. And you see that there are many more number of positively charged residues, percentage. Red is moved toward the right. So you have more positively charged residues at the inside. Okay? This one here, more blue in than out. More positively charged. If that is the case, then all you now need to do is you predict the helices, count the positive charges on either end, and assign. Gunnar did an additional step. So he was the one who discovered that rule, and actually has done very recently a lot of experimental evidence that explains why this rule is the case, why this is actually true. And his idea was then, since that seems to be such a firm rule, you could use that to predict the membrane helices. How would you do that? So essentially the idea is you move the threshold around so that the model, the number of membrane helices, is optimal for getting the difference between positive inside, so positively charged residues at the inside and outside. It's a simple additional thing to optimize, and that is his method that he used. Here's another method. Before we get into that other method, I wanted to dwell again on, and I just put back Gunnar's face because it's somebody I'm very impressed by. He has most likely done more to the membrane field than anybody else. So here's another experiment that I said that membrane helices typically need 17 consecutive residues in a helix to pass. Now, assume you have one long helix, and let's assume that you just add residues to this that are propensity for forming alpha helices. Let's say you duplicate that helix here. What will happen? So you make the helix longer and longer and longer. What will happen?
Could be a long loop or could go back inside.
It will go back inside. And so what he now experimentally analyzed is, what is it that you have to put in between to induce a kink to go back inside? Or how long does the helix have to be? And it turns out that if you, I can't remember, I should show you the number, I'm telling you just as a story, it's about 30-something, 34 or 35 residues. A helix without any break will just go back in. If you make the helix long enough, will just go fold itself and go back into the membrane. If you put the particular residues in between, you can make something that is shorter, force something that is shorter to go back in. You create a kink.
Are there proteins that go, like, do this inside the membranes that don't even go out?
Yes, we do have them proteins. And in fact, Gunnar has an example. It talked about an example that is looking like one of the structures I showed you. Let me get back into the structures. Where was it? I'm sorry for the animation now. Oh God, where did I had it? I just saw it. By the way, that is the one that really gets ions through, a potassium channel. No, I'm sorry, it is somewhere, but I lose it. So here's an example where that is the membrane. That is something that is predicted by methods as a membrane helix, and it is in the main part of the protein. It is not in the membrane helix. And what he figured out after a few years of doing experimentation is that in fact there is some condition by which this would be inserted in the membrane. There's some native condition of this protein by which the region that in another native condition, I can't remember the details of the story, the helix is part hanging out in a way that energetically seems to be unfavorable. It's not even really in the core of the protein. It's just sort of dangling around there. It's predicted by all methods to be a membrane helix, and for years he couldn't observe it. Then he found another experimental condition, a multiple maybe binding maybe multimerization, in which this particular helix was inserted and was a functional membrane helix. Is that answering your question? Was that what you asked? In some sense, it's an extreme answer to your question, right? We have examples, and again, I believe I showed you some, where you have a membrane helix going through the membrane, and then if that is the membrane, I have a long membrane helix, and here it goes into the globular part of the protein. Inside of the globular part of the protein, the helix is a helix in the hydrophobic core of the protein. And it passes through as one long helix. Still not what you asked.
I would ask, can a protein make a loop and then
Yes.
not have a kink but go back without having it? Like do this one in the membrane without he's a membrane and doing this one inside it. So looping inside it without once.
I do not believe we have the so-called re-entry helices, and we will
Right. That's not re-entry. Like this looping inside without going
Yes.
in the membrane.
Yes. That's a re-entry helix, right? We talk about. So essentially, imagine I would be strong enough to put my fingers in. These two fingers are just one long membrane helix,
Right.
and as one long membrane helix. Oh God, that is the long membrane helix, and as this long membrane helix, you get through the membrane. And instead, now I put them reaching half through the membrane, and it goes back in. Like this.
Yeah.
Is that what you meant? So this is outside,
Yeah.
and they're both sitting in the membrane,
Right.
and they're both half of 17. So both like 9 residues, or 6 to 7 residues. Is that what you meant?
I think so.
That does exist. It's called a re-entry helix. I'm not aware of any case, we have many of those. I will possibly gloss over a few images from those later, but I'm not aware of any case where we have in, out. So one in and out, yes. But so you imagine your finger kept going. It's not snaking back in and out. Okay, so there are some additional complications to membrane helices, and a lot of the experimental work for that has been done and is being done by Gunnar van Heijnen. Let's just, I want to make one point on this one here. From the lab of Stephen White comes an idea that simply says, okay, there are different hydrophobicity scales out there. What I wanted to use the hydrophobicity scale for is to predict membrane helices. Why not optimize the hydrophobicity scale such that I can predict membrane helices best? And that's the idea of that method, and it works fairly well. Now let's get back to this issue here. Polyvaline, so this particular stretch that is completely predicted as a beta strand. What else could we do in terms of hydrophobicity? Well, we could use the same kind of approach that we had before, where we predict secondary structure through, in this particular case, a machine learning device, and feed possibly a profile to improve it with a profile. Now we can translate the whole thing into the membrane story. So instead of having 3 output units, we have now 2. Membrane, not membrane, single sequence input, profile input, same thing as before. And then we have 2 layers, because the first layer in this particular case of neural network had helices too short, so we had a second layer. Now, if we did that, then the second layer now brings in a problem that makes helices too long. So you have to cut them again. Cutting them again, one way of doing that is by some sort of dynamic programming in which you perceive the output unit for non-membrane as an energy landscape. Here's the residue running, so sequence residue number 1 to, that's one particular protein here, to the last, and that is the energy landscape for a transmembrane unit. You see there are one minus of the other, of each other, these two. So when this one is high probability predict membrane helices, this is low, and vice versa. And now you simply find the best path through that energy landscape, if you want, that optimizes the value that you get given simple constraints. You know that membrane helices are between 15 and 25 residues long. So you do the following. You label all your segments in your protein according to that energy landscape between 15 and 25 residues, and give an average for each of these segments. Now, if your protein has one membrane helix, you simply pick the segment with the highest number. This chosen example is that one. If you assume that the protein has two membrane helices, then you pick the second highest. And this one, in this particular case here, the second highest is here. That you cannot pick because it's the same that you already had before. So if you predict one membrane helix, that is your best prediction, given these initial segments here. So the second one cannot be here, but it must be somewhere where you have no overlap, where you have enough in-between membrane kind of region, and that in this particular case would be here. So if you predict 2, then these 2 would be your best prediction. And you can build up. And then at some point you have to decide whether adding another one gains. And that is simple to do because essentially every one of these is a probability of forming. So when what you add is below 50%, so when the probability of adding another segment is more likely to be not membrane, you stop adding. And that way you build a model for which you know that given the number of helices in this particular case 3, if you predict that in this particular case the model predicts 3 membrane helices that are optimally compatible with the idea that you have the landscape here, energy landscape of predicted membrane helices, and a sort of limited number of length, right? Very simple algorithm. And then you simply use the positive inside rule, and this way can predict membrane helix. In this particular case, the method predicts this. Now, here is an example I briefly mentioned in the context of alignment methods, hidden Markov models. So this is transmembrane helix predicted hidden Markov model that originally came from Anna Schroch. Some of you may remember that he actually brought also hidden Markov models into the field of alignments. Here he paired up with Eric Sonnemer and Gunnar van Heije to develop this method TM-HMM. The beauty of hidden Markov models is that you can put some grammar into the story. And the grammar here is simply, you have some reality of the membrane. You have a helix beginning, a helix core, a helix end. You have some reality of the outside. You have some reality of the inside. And all of that you can model easily into different states. So somehow grammar is built into the membrane protein. In a globular protein, this is not at all the case. The helices go in and out wildly. And there is no alternation. So some things such as a timbarrel, for instance, has certain alternation motifs. But there is no general alpha follows beta or alpha beta is like this. Nothing like that. In the membrane it is exactly like that because it can only be alternating. Membrane helix is always followed by non-membrane helix. Cannot be 2 membrane helices. Can, but it's a different story. Anyway, so it's a limit of that model, let's put it like that. TM-HMM, very successful first. Here is the second one, Isvan Shimon, Gabor Shnadi. A similar idea, again, this one is called HMM top. And the difference between these two, TM-HMM and HMM top, is simply in the way you define the microstates in between. In many ways that implies the way you define the grammar. This is slightly different, and the way they filled it with data is also different. So in order to do that, they developed their own database. But in principle, the idea is similar, and they benefit from the same strengths. Now, I'm not very sure how to zip through whatever I wanted to zip through. This is going to be a fast forward. So at some point, I and others developed methods, and we claimed a certain level of performance. So my next question, I presented methods to you. Now the question is, how well do they work? And here's an answer that was given a few years ago, 96, so almost 20 years ago. Let's not look at the numbers because this is totally turned out to be totally wrong. And there are many reasons why this turned out to be wrong. Ultimately,
how do they even validate it without, and so less of testing data? How could they validate without like?
So I tried to publish a paper in Protein Science 6 years later, saying we were utterly wrong, and we got totally beaten up. This manuscript got taken apart. I mean, most of my papers did not sail through. Very few of my papers got through clear. Most of them are somehow have had problems. That was the peak of all the problems we ever had. And we absolutely got killed, and in such an aggressive way. And the reason why we got killed, I had never realized that. So what I attacked is a paper that I had written. I attacked myself. I said I was an idiot. I was totally wrong. But I also attacked all the others who claimed the same. I had not thought about that. I was a leader in the field, and by attacking myself, I attacked everyone else. And all the reviewers felt attacked. They hit back. I could never publish that paper. I published something, so actually we made one paper out, we cut it down and we made 3 papers out of it. We published 3 papers out of it, but none of them really made the statement we were wrong. They all looked at a different element of what went wrong. The major thing is exactly what was done before. Biochemical labeling shows you whether that particular residue is outside or inside of the membrane. This, when we started to come up with these numbers, we had essentially four crystal structures available. Hundreds of proteins, I don't remember where, 130 proteins were used to assess these numbers, and most of these then, 131 minus 4, were done by low resolution experiments, or by labeling experiments. Very carefully done. Those days, each one of these is a PhD thesis. So they were really carefully done. And what we showed also in this paper is these low resolution experiments were worse than prediction methods. So that's in some sense the answer in 2 ways to your question. How could we do it? Because we believed we had good experimental information. We found out it was not good, and that's how we fooled ourselves. Or how the experimentalists fooled us, or whatever you say. That was the other part, what I could not easily publish, because ultimately the great story, I told the story as a loser story. But the success story of the part here is that on 4 real crystal structures, the methods were good enough to be as good as all the experimental low resolution methods, on average. That's remarkable, if you think about it. That's an amazing success story of computational biology. But that was also not easy to publish, because there were reviewers who earned the bread and butter from the experiments. Any question on this part of the part that is offline?
Yeah.
I can see it. So here's a paper from Jonas Reip. And so the first question that he comes is, when Jonas was sitting where you sit, so he was in one of these courses, and then our exercise then was to assess how well membrane prediction methods work, and that got into a longer story. So which methods would you use to assess how well the methods do? All is not a feasible answer. One is because there are too many that are only hydrophobicity based, and there are too many scales. There are over 30 sort of advanced methods. Should we also actually use consensus meta and meta methods? How should we choose which one? How would you choose it to assess? So I give you the task to assess membrane prediction methods. Too many are available. You cannot choose them all. How would you choose them?
Choosing some kind of groups, some kind of
types of methods. That's a great solution. What else could you do? That is an aspect that will be on the next slide. What else could you do? Like, think simple.
Choose random, just a few.
Yes, but that could be unfair. That's true. So this actually gets you into two problems. One, it could be unfair. It was my immediate answer. The other thing, you have to always build credibility with a reviewer if you ever want to publish that, so that they believe you that it was random and that you didn't choose the worst ones or whatever is relevant in your message. So that is a dangerous choice. It's much simpler, guys. Well, so it's very difficult to make methods run. So clearly, one thing that you want is that the method, you can somehow access it. One thing that you want is that people still use it. That the method somehow is common, that the method is somehow attractive, that people know about that method. So the idea of orthogonality, so unusual, is also important. Somehow recent methods clearly should be in. When you publish a method today, you always have to somehow make the point that if it's something that people did before you, that you at least do add something new. Either a new method, or you do better, or you add a new thing that can be done with your method that couldn't be done before. All of that points to the point that you should clearly look at recent methods. Now, you always need a perspective. If you analyze prediction methods, you need two levels of perspective. One is what is random, on the lower end of things. On the upper end of things, what is where do you get a perspective from? How well can you do? Well, there are two ways of doing that in the world here of protein structure prediction. One is you look at structures that are done twice in two different labs. How do they agree in terms of the thing that we measure? Here is the number of whether residues in a membrane or not, or membrane helices. That's what we're trying to measure here. Immediately you would ask for proteins that are same sequence, done twice, how much do they agree? And clearly you cannot have a prediction method that is better than the experimental agreement. Or, now we get into comparative modeling. We are predicting 1D structure because we cannot do comparative modeling. If we can do comparative modeling, nobody cares about this. If we can predict the 3D structure of a membrane protein, nobody is going to turn to secondary structure prediction. So, actually, in some sense, the ceiling here is the degree to which similar proteins, you take two proteins that have a similar structure in terms of 3D, count how much of the membrane helices agree. That's another ceiling. But let's get back to the random. So one thing that you could do as random is you randomly predict a residue to be in a membrane or not. What else could you do to sort of measure a random background? I'm looking at you. There's a reason, but I'm not going to tell you why. Only afterwards. No? So the reason is you came with this idea that you look at the distribution of lengths of secondary structure segments as a secondary structure prediction method. I told you for globular proteins it doesn't work. For membrane proteins that exactly works, right? Because you have a fairly, helices have a distribution that is much helices and globular proteins have a distribution that is much wider than the distribution of the length of helices and membrane proteins. That's much sharper. And there it's much simpler to use that very idea and to come up with a way to use that idea. Here, for instance, to a random prediction, you could in fact predict segments of fixed length that you simulate from the distribution you have. And you just pick the point, right? It's still random, but sort of a more clever random. And a more right random in some sense. Then you could also, for instance, adjust the number of segments. So you know that on average you have a certain distribution of proteins of 1, 2, 3, 4, 5. And that background you can also use to simulate here. So all of that still is random. You pick ultimately the segment at random. But now you pick a distribution that if you looked at the random distribution, you would not get helices of length 1. Which is nonsense for a random method. So what you would create with this is something that overall, in a simple scale, looks like the observation and is still random. Here is sort of what Jonas called semi-random, where you look up from the experiment how many membrane helices you have in that protein, but you randomly place them. So that essentially measures if somebody knew the number of membrane helices. How surprising is it that you get them right? Okay? And then there is the final background, and that final background needs some explanation here. Hydrophobicity essentially is the first part of the idea of Gunnar von Heijne, needs some explanation because ultimately Gunnar von Heijne did this idea fairly elaborately 20-something years ago. So if people have evolved methods over 20 years that are more intelligent, then this is at least what they should beat. In that sense, this is also, in some sense, a random background. Actually, I'm going to show you Gunnar's method too. And this really is a trivial, stupid, or simplistic way of doing random. The data set to choose, we have very few structures, but the problem with low resolution I talked about is we have a lot of data here, but it's not good enough, so we really need to use the few structures we have. But in the few structures we have, here is shown bacteriodopsin, there is one problem. So that is a structure of a membrane protein. Do you know what the problem is when I see the crystal? Well, where's the membrane? You don't see the lipid in the structure. Why not? Is it because there's no diffraction from a lipid? No. Okay.
Isolated? Weren't isolated?
Yes, you're absolutely right. So the problem is they sit natively in the membrane. In order to grow crystals, you move them out of the membrane. You do sometimes have a surrogate. You put them in an environment that somehow is enriched in oily molecules, and that is what you sometimes see, but you absolutely do not see the natural native lipid. Although you don't see the native lipid, do you have an idea if I told you that the membrane helices are orthogonal to the membrane? Do you see the membrane? I don't see it, but is there anything that sort of would, if I brought you to the whiteboard and asked you, show me where the membrane is, would you stand there and say, I have no idea? Or would you sort of, you have an idea, right? It's remarkable how you can immediately see here that in fact this is sort of, if you want a membrane like that, you would have to rotate it. You immediately see that this orientation is not compatible with the idea that the membrane runs here. You do see that there is somewhere a level like this. You see many things from a structure that does not actually contain the lipid, but there is some room for change. So there clearly is not a well-defined end and beginning. And the way these structures are assigned from 3D coordinates to membrane helices, the two methods here, OPM and PDB-TM, essentially they look at this, what people refer to as the hydrophobic cuff. So there's some sequence signal that you see at the ends. Jonas used both methods and said a method is right whenever it agreed with either of these two assignment methods. In this particular case here, you see the blue one, I lost that, it must be inside and that is outside. So the red one is the residues in the outside of the membrane, the blue one is the residues outside of the membrane, yellow is inside of the membrane. Now, the question is, how do you measure accuracy? The simplest way is the Q2 as a percentage of observed. So how many of the residues here that you predict to be in the membrane are predicted correctly of all the ones that are observed and of all the ones that are predicted, often recall and precision. Whoops, the answer here for a bunch of methods, where the newer ones are more on this side here, the bluer side, not completely true. MEMSAT SVM is one of the newer ones, and those are the different semi-random. Remember, somebody gives you the number of membrane helices and you randomly place it, hydrophobicity. So that's the background. The first observation, totally random, is worse than the methods, much worse. Second observation, trivial use of hydrophobicity is fairly accurate. Third observation is that they are all doing fairly well, but they are not easy to distinguish. Again, this percentage of observed, percentage of predicted. The field is very closely together. Now, what we are looking at here is whether or not particular residue is predicted correctly in a membrane. I told you that the assignment of the membrane is not entirely well defined. You certainly can move plus minus one residue. You don't see it in the crystal. So if you have a 70M protein, plus minus one immediately on both sides immediately gives you 7 times 4, already 28. And a protein of 300 residues, that is 10% difference right there, where methods sort of reach almost 90%. 10% mistake in the assignment is a big step, right? So maybe that's not the right way of looking at it. What else could we look at? And here's another way. This slide is just making the point. What else could we look at? What makes more sense? I'm not sure that I get to the re-entry helices. I'm some distance away from my re-entry slide. So I will continue on these slides anyway on Thursday. But I do want to get into the beta membrane story. So I'm not sure. But anyway, so this question I still like an answer for. Not measure single residue. What could we actually measure?
Segments.
Segments, exactly. So we could look at the number of segments that we correctly assign. Now, when we assign a segment correctly, we get into the next issue. The next issue is you will have an observed membrane helix and a predicted membrane helix. In this particular case, it's totally trivial. The 100% overlap is right. But when do you say that you have the segment correctly predicted? How much leeway, how much mistake do you allow? How would you choose that? Well, here's one solution. How would you come up with your own? Or, well, okay. One absolutely fair answer is to say I would look at Google what others did. In this particular case, historically, people had often used the number 5 overlap. So saying that as long as they overlap by 5 residues, that was enough. And here's an additional problem that people made. So here's the observed helix. Say that would be the predicted helix. And that would be 2 observed helices. And they double counted. Saying that, you have more than 5 here, you have more than 5 here, so that's 100%. Or in a case like this, well, that is also 2 times 100%. So 100% correctly predicted. You could also look at it and say this is totally wrong. Because a membrane helix has to be at least sort of 17 residues. So an overlap here by 5 and here by 5 within so you get the number wrong. You don't really get the core of the helix right. Most of the helix is wrong. Most of your prediction is wrong. So calling that a 100% right prediction is not really right. But that is what you would have found in most of your Google effort. So this is a unique stance that says this is nonsense. Although the entire literature gave this, of course, Jonas was helped by having me in his group. So I had been responsible for many of these mistakes, and then it's easy to correct them if you make them yourself. But again, the question is, how do you? I understand we completed this a minute ago, but the question remains: how do you choose the number?